Newswise DALLAS, March 3, 2023. Cell membrane protrusions called BlebS, which typically mark the end of life for healthy cells, do the opposite for melanoma cells, activating processes in these cells that help them survive and spread. According to a study at UT Southwestern, the findings, Nature, could lead to new ways to fight melanoma and potentially a wide variety of other cancers. Our old adage in biology is that form follows function. But here, we're reversing that concept. We show that by changing form, you can change the chemical processes inside the cell that control their function, he said. Dr. Danuser, Bioinformatics Instructor Ph.D., co-directed with Andrew D. Weems at the Danuser lab. Weems explained that if healthy cells detached from larger tissues are unable to reconnect within hours, they face almost certain through a process called anoikis. However, one hallmarks of malignant cells is their ability to survive indefinitely after leaving the tumor tissue, allowing them survive and migrate other parts body form metastatic tumors. While detached, they form bubbles indefinitely, whereas healthy cells can only form bubbles for about an hour after they leave their tissues of origin. Previous research in the Danuser lab has shown that bubbles collect proteins that can promote cell survival under stressful conditions. But whether these proteins help cancer cells avoid anoikis and how these proteins are found in the bubbles is unknown. These investigations showed that the BlebS attract a family of proteins known as septins, which are attracted to areas of high cell membrane curvature, such as those found in BlebS, forming molecular scaffolds that provide a framework for proteins to interact. Here, septin scaffolds contain ROS proteins that regulate cell survival and are mutated in about 1/5 of all cancers. Weems said, when researchers treated melanoma cells with a compound called forchlorfenuron, which inhibits septins were no longer able to scaffold ROS proteins, causing the cells to be unable to sustain chemical reactions that kept them alive. So, when they separated from other cells, which was the same fate as healthy cells, they, conversely, when they introduced mutations that caused healthy to bubble longer than normal, these cells became as anoikis resistant as cancer cells, even though these did not harbor cancerous ROS mutations. A member of the Harold C. Simmons Comprehensive Cancer, Dr. These findings suggest that pseudo-oncogenic mutations alone are not sufficient to drive cancer functions, but may depend on specific cellular configurations to do so, said Danuser, UTSW Center and Texas Institute for Cancer Prevention and Research Fellow. On the other hand, promoting these cellular configurations alone can activate cancer-causing functions incompletely healthy cells that have not been mutated. This offers a fundamentally new perspective on cancer development. Researchers have tried to inhibit ROS activity for decades but have been unsuccessful. Weems said, ROS itself is considered impregnable due to its unique molecular structure. He added that by targeting septins instead, researchers could achieve same goal, removing ability of cancer cells to resist anoikis, potentially making them more vulnerable to other treatments such as chemotherapy, radiation or immunotherapy. FCF has been used as an agrochemical in the United States for nearly 20 years. And Environmental Protection Agency data indicate low toxicity to humans, Weems said. This septin-inhibiting compound and its chemical derivatives could be promising candidates for new anti-cancer treatments. Dr. Danuser holds the Patrick E. Haggerty Distinguished Chair in Basic Biomedical Science. Dr. Weems is a member of the Jane Coffin Child's Memorial Fund. Other UTSW researchers involved in this study include Erik S. Welf, Meghan K. Driscoll, Felix Zhou, Hanieh Mazloom-Farsibaf, Bo-Jui Chang, Vasanth S. Gabriel M. Gihana, Byron G. Weiss, Joseph Kai, Divya Rajendran, Kevin M. Dean and Reto Fiolka. This work was funded by grants from the National Institutes of Health and the Welch Foundation. The University Texas Board of Trustees has applied for a patent on the therapeutic targeting septins and BlebS, based in part on data from this study. Dr. Danuser and Weems, Dr. Welf and Driscoll are called inventors. About UT Southwest Medical Center is one of the nation's leading academic medical centers. UT Southwestern integrates leading-edge biomedical research with exceptional clinical care and education. The institution's faculty has received 6 Nobel Prizes and includes 24 members the National Academy Sciences, 18 members the National Academy Medicine, and 14 Howard Hughes Medical Institute investigators. More than 2,900 full-time faculty members are responsible for groundbreaking medical advances and are committed to rapidly translating science-based research into new clinical treatments. UT Southwest physicians care for 100,000 inpatients, more than 360,000 emergency room cases in 80 specialties, and oversee approximately 4 million outpatient visits annually.